Welcome back, everybody, from the break. Our next speaker is joining online from Chicago. It's Professor Emeritus Dr. Todd Kuiken. Thanks a lot for joining us today, and we can proceed with your presentation now.
So we're going to talk about TMR for prevention and treatment of amputation pain, not for prosthetic control, as I usually discuss. I want to highlight that this work has been done by dozens of people over the last 20 years, including in our lab, the Center for Bionic Medicine, at the hospital, Northwestern Memorial Hospital, and in collaboration with some of the engineering and surgery departments. Quick disclosure, I'm part of a company that developed a control system for prostheses. I don't think I'm going to talk about it much, so I probably don't need that one. So I've spent most of my career, 20 odd years of it, at the old RIC. About 3 years ago, they made a new building, 3 times the size, very pretty, and they did it in a very new scientific model. What they've done is on all the inpatient floors, the labs or the gymnasium are shared. So you'll have all the patients walking around with tracks and all kinds of gizmos while the scientists are doing their things in the middle of it all. It's kind of noisy, but it's interesting. So Levi Hargrove is now the director of the Center for Bionic Medicine. This is him, I don't know when, back at the Turin meeting a long time ago. We've been working together for about 20 years, and he owns the place. It's no longer my fault. I retired in February of this year, guys. And here was part of the team in 2017. We have about 60 engineers and clinical staff, and then a few graduates and postdocs and writers, and it's a really great group of people. Very much transitionally oriented research. So, as I said, there are 2 types of research. One for improving the control of prostheses, and number 2 is for prevention and reduction of amputation pain. And they are different surgical procedures with different goals. So I'm going to talk about number 2 first because, we're at a pain conference. An extra shout out to Greg Debanian. He's been my surgical colleague for those 20 years and helped figure all this stuff out. So again, the list of people, like you're going to remember all those names. I'm going to highlight Eric here. It looks like Max has borrowed Eric after he finished his PhD and is working in Chalmers now. Max, I'm very impressed that you guys did this meeting. Small meetings like this are really, really hard and take a ton of time, especially when you don't have a national society or somebody supporting you. Doing them off on your own is not only hard, but it's also kind of risky because you got to get enough people to break even, and nobody will sign up for the meeting until the last month, no matter how hard you try. So Max and guys, good work. I think this is a great topic for the meeting, and I appreciate your hard work. Okay, treatment for neuromas. So every nerve that gets cut develops a neuroma. We only worry about them when they're painful, but every nerve develops one because it's what it's supposed to do. The nerve gets cut and it starts doing some sprouting so that it has more nerve fibers to go out there and try and reinnervate something. But typically it forms a little ball of scar tissue and a tangle of the axon ends. And often this is hurtful. It turns out that TMR reduces both neuroma pain and phantom pain. And this is a very simple hypothesis for why. So if you cut the nerve and you give it something to do, it's happier. So the nerve is cut, and you cut a nerve to a little nerve to a piece of innervated muscle, and now this muscle is denervated, and the axons will regenerate down into that muscle and form at least some functional connections. And so glass half full, you're making the nerve happy. The other way to look at it is glass half empty is a typical nerve that's cut is a angry nerve, a pissed off nerve because it's not working right. So here's a little bit of data. This is some work our plastic surgery residents did in a rabbit model. So here you can see a normal nerve, and I think most people agree those are pretty nice looking cells with their Schwann sheaths and everything. And this is a regular neuroma where you don't see much of anything except a lot of fibrous tissue, a few axons in there, and it's a mess. And if you do TMR, it's something in between. These nerves are not as healthy looking as normal, but they're better than the neuroma one. So, and you also have less sprouting when you do the TMR. So this started to give us some scientific evidence that, you know, TMR might work for pain. So I had been hearing from patients every now and then that TMR had helped their phantom limb pain or their neuroma pain, and just here and there. And well, you know, that's nice, but a casual comment from a patient or few is not very compelling to me because pain is inherently so subjective. You know, and our research subjects we work with closely, we like each other, and they want to make us happy and tell us nice things. So I had trouble making too much out of it. Oh, here's a tiny bit more data just showing how the fiber counts after TMR go down compared to a neuroma. So anyhow, we added this rabbit data, and now we had a number of patients saying they felt better along with some real data in an animal model that could give you a kind of a reason why. So we did a chart review. And so we had 28 patients with a total of 15 painful neuromas before we went in and did TMR. And after TMR, all of these neuromas were better, but one was still painful enough that the patient had trouble wearing his socket. Now, there were 2 new neuromas, but they were on nerves that we never touched because, you know, these are different levels. And in the shoulder dysartic, we're usually using all 4 nerves. Transhumeral, we're doing a couple of nerves, maybe 3. And so you could have a spare nerve form a neuroma. So this was, again, nice, another piece of information, but a retrospective chart review is not going to make this paper go to JAMA. So we got a great big Navy grant to do a very well done prospective study. Greg was a PI, and the paper came out in Annals of Surgery 2020. So what we did is recruited amputees, and it was a blinded study that they went into the operating room not knowing which procedure they were going to get. The standard procedure, which is just to cut the nerve back, or TMR, which is cutting the nerve back and sewing it onto another small nerve, generally over the motor point of that small nerve. So we thought about blinding the surgeons too, but we thought that might interfere with technique a little bit. So in this paper, might as well just go to results here as opposed to pages of them. Their phantom limb pain scores went down and had a clinical significantly different. Maybe I do have a bunch of slides. No, I don't. I'll have to stick with this. Essentially, the pain for both neuroma and phantom limb both went down if the patient was already an amputee having that pain and they were then reoperated on. And it was done on a series, it's a different paper, but it was also done on a series of new amputees so that to see how much phantom pain and neuroma pain they developed. And if they had TMR, they did better than the control of the standard procedure. All right, so that's TMR for pain. The difference I'm going to highlight in surgery without a bunch of slides is when you're doing it for pain, the surgeon basically doing an amputation, and they got a few big nerves that they would like to treat. And what they do is they find a little nerve going into a piece of muscle. You can even see my fingers now. They find a little nerve going into a piece of muscle, cut it right where it goes into the muscle, and then they take the big nerve and sew it right over that innervation point, which is easy. You just throw in 1 or 2 little stitches, and the regenerating nerve then has a track of degenerating axons, which kind of sucks it in, if you would, and that nerve can reinnervate that little piece of muscle, and that relieves and prevents pain. Exactly why, I don't know. I mean, you got right off the bat, my very basic making a nerve happy or not by giving it something to do. I hope someday someone figures out a little more detail than that, but I'm not smart enough to do that. And I'm retired, so I don't have to. All right. Now, when you do TMR surgery for amputee pain, the goal is to give the amputated nerve some denervated muscle to reinnervate. It's very important that you understand that it's denervated. You got to cut that little nerve. You can't just sew it next to it or it won't work. Nerves will only grow into denervated muscle for the most part. There was a paper some 30 years ago by some silly graduate student that looked at reinnervation with muscle with and without denervation and a few other variables that you can read. Had a weird name. I think it was Kuiken. So this surprises me. The size of the muscle being reinnervated does not appear to be a key factor on giving this pain relief. I would think because you got this big damn nerve the size of a pencil and you're going into a distal nerve the size of pencil lead, that there might not just be enough nerves to do it. But so far it seems that, at least as far as I know, there is been no one's appreciated the muscle size being a huge factor. Maybe Paul can talk a little bit about that more if you guys want. The surgeon doing this, they can find pretty much any little motor point they want because they're cutting a small nerve. It's not going to affect function. They're already in an amputated limb, so there's no function, even if there's no function because the arm's gone. So they can pick pretty much anything they want and sew them together. And so those are the keys for that. Now we're going to go back very briefly and give my talk that I used to take an hour for and have been giving for 20 years. So that's the idea of using nerve transfer surgery for improved control of robotic prosthetic limbs. So you lose your limb, your muscles and bones are gone, but your nerves are still there and they're still alive. And you can tap on the nerve of a World War II veteran and they will still feel their missing body part much of the time. And if we want to control our arms better, we need some kind of biological amplifier that will get us info on what to do with this robotic arm and all those joints, wrists, fingers, all that. Right now, the state of the art is we're lucky if we get good control of hand open and closed, wrist rotation, and elbow. But if we add the 4 signals from the brachial plexus, you've got 4 new signals to extract information from. And so at least at a transhumeral level, without any fancy math, you have one head of your biceps left to do elbow flexion. You got your triceps extender, and then with the TMR, you got a hand flexor and a hand extensor. So you can control the elbow and the hand independently and at the same time, which is pretty cool. Now, if you add pattern recognition, which is a way of collecting a bunch of signals and then correlating the pattern of EMG firing to what function the amputee wants to do, you can get even better control. And that's been a very exciting path and fun to watch it grow. So by moving these amputated nerves, here you see an example with the pec. It seems to be the most easy for a shoulder dysartic. You basically get an amplifier. The muscle is a biological amplifier for the nerve signal. And for TMR, for improving prosthetic control, it's not just any nerve anywhere, because it does matter how big the muscle is. You want the muscle to be big enough to be a good amplifier. And it seems to be that there are certain strategies that work better than others. So there's definitely room for wiggle. And I've seen some, shall we say, interesting deviations of this plan. One time a guy lost his arm and he had a big tissue deficit up under his clavicle. So the surgeon used the serratus muscle down on his side, which has got like fingers of muscle, and put it up there to cover the open spot, and put one nerve into each of these little fingers of muscle, and then put split skin graft over that. And I saw that patient in the clinic, and I'm going, and he could make the little spots fire, but I was looking at little twitches, like someone give me my more powerful glasses. I could barely see this. But it turned out to work pretty good for the prosthetist, and I think it's because of the split split skin graft. There was such little thin over it. The electrodes were right on those little pieces of muscle, and he was able to control the prosthesis pretty good with a clever fitting prosthesis that our prosthetist made. So our very first patient was Jesse Sullivan, just a saint of a man, great guy. He was 54 years old at the time that, as a lineman, he touched the wrong wire, had a 72,000 volt burn that burned away both of his arms, and by all rights should have killed him, but he survived and had immediate bilateral shoulder disarticulations. He came to us for rehab, and one of the things we noticed was he had these split thickness skin grafts up here in his armpits, and they hurt. Just taking a shower, having the water on him hurt. So he needed those fixed. And so we were just at the time to do our first TMR, which we were planning for transhumeral, because that's ideally a better level. But anyhow, we had this great guy here who needed surgery anyway. So we asked him if we could rewire him while we closed these wounds. He said, "Sure, doc." And his insurance company said, "Okay, guys, you can do that." And thus Jesse was the first TMR patient pretty much fully paid for by insurance, which was nice. Insurance companies are not all evil. So here you can see kind of the surgical plan again. The pec is actually not one muscle, it's at least 3 muscles, and you could make an argument that it's almost an infinite number of muscles because of the fibers just running out either laterally or up and laterally. So this is what we use. Now the pec minor is generally underneath the pec major, and so Dr. Demanian kind of moved it over laterally and used that for what we considered the least important nerve at the time, the ulnar. And when we did the surgery, the muscle got a little purple, which was concerning, but Dr. Demanian thought we would be okay and it would survive. You know, I'm going to do just 1 or 2 more slides and quit so we can talk. I'll go back. Machine. No. Back. Oh, well, I guess you didn't want to see that other stuff anyhow. Because I'm not smart enough to figure out how to go backwards on this machine. Okay.
Relax, close, relax, open, relax, close, relax, elbow up, relax, elbow down. Relax,
So this is Jesse's chest as he thinks about closing his hand and flexing his elbow. I'll challenge anybody at your meeting to be able to do this with your chest. And if you can, Max owes you a big Swedish beer. Fascinating how you can see we really got pretty nice distribution. It's not just one big mess of nerves. We got we got 4 different places you could pretty reliably count on for the different movements. And the median nerve even spread out over this planar muscle a little bit more. There was a spot where if he did his pinky finger, you could see the muscle twitch. But it's not like I could put an electrode there and control a pinky finger because I didn't have a pinky finger motor to control. So with these 4 sites, you could control a 2 degree of freedom arm, both the elbow and the hand at the same time. But if you put the pattern recognition in, which decodes the muscles better, Jesse's been able to control up to a 6 degree of freedom arm. And unfortunately, that's a whole nother talk, and I don't have time, so I'll just quit. Oh, and here's Jesse. Same arm before and after surgery. So this is his arm before TMR, and he's using little switches up in here to control it. And here he is using his electrodes, which was quite a challenge. Keeping, now you have, I think we had 6 or 8 electrodes in here, and you got to keep them in the same place. But, as you wiggle around and move and sweat, those were all big challenges. But he did great. And I'll tell you, you'll never get a better spokesman for your science than Jesse. He was such a powerful man. Okay, now if I can figure out how to turn this off, we can do questions.
Thank you very much, Todd. So we have a couple of questions. You probably hear the applauses.
So I didn't hear it.
Well, everybody applauded loudly.
Oh, thank you.
One of the questions is, in your Annals of Surgery study, there appeared to be a large difference in baseline of PLP between the active and the control groups. Could this explain your findings? So the difference in the baseline.
No, I can't. I mean, everything was randomized from the start. You know, the patients were randomized and coming into it, and no one knew which category they'd fall into until the surgeon opened a little envelope in surgery. So I can't think of a good reason why there would be a difference in the population.
Okay.
Sorry.
And the second question reads, how does TMR compare with other, arguably less elaborate surgical nerve transfer techniques used to treat peripheral nerve injuries with respect to PLP?
Well, I'm going to let Paul talk more about that. He has a technique that I don't know if it's less invasive, but it's different. And so what's kind of cool is so we invented TMR as a concept. You know, it's not like you got to do ABC for the most part. We invented the concept. And surgeons don't like to do things the same way twice. It just, I think it's something that gets into them that, as being boring or something. And so several surgeons have come up with new ideas on how to get information for control of prostheses or variations of this technique. Oscar Obmann in Vienna is kind of taken the technique almost to an extreme by trying to harvest nerves farther down below the elbow and reconnecting nerves down where in ways that haven't been done. Other people have done some pretty interesting targeted sensory reinnervation surgeries. We did the first targeted sensory reinnervation surgery, but there are a number of ways to do that. And Jackie Hebert did a rather elegant technique up in Canada. So I look at this more as a concept rather than a specific surgery.
Okay, thank you. The concept as a nerve transfer, I suppose.
In a way where you're using the parts differently. You know, nerve
Right.
transfers are usually used to make a biomechanical movement go better. And we don't care about the biomechanics. We're caring about, you know, the muscle signal, which is very different. So I kept saying how for pain, the surgeon can use pretty small pieces of muscle, but here you're looking at a transhumeral slide, which is what we use for transhumeral amputees. And I want big amplifiers. You know, I don't want little muscles, because if I got a little muscle, it's hard to pick up the EMG signal on the surface. For this kind of TMR, I want a more standardized procedure and big fat muscles and not a lot of fat around them, because the fat's an insulator. So I don't have it on in this slideshow, but we did surgeries where we all have a fair amount of fat in our arms. The arms in our belly are apparently where we store a lot of fat. So rather than remove a bunch of this subcutaneous fat, we put it between the heads of muscles. And it helped to give us some spatial separation and made getting signals better. So it's a matter of looking at the physics of the problem in a different way.
Okay. Thank you very much, Todd. So now we're gonna, I hope you can stay for the panel discussion in about an hour. Or if you go, you can come back. Thank you.
Right now, I gotta see if I can get back to functional here.
It's good. So now we